Hello everyone. In this video, we discuss the basic introduction of chromatography. First of all, look at to the history of chromatography. Michael Twisty, a Russian botanist in 1936, used chromatography to separate plants' pigments. He called the new technique chromatography because the result of analysis was written in color along the length of the adsorbent column. The basic meaning of chromatography is chroma mean is actually color and the graphian means to write. The importance of chromatography is very broad. Chromatography has application in every branch of physical and biological science. Uh, 12 Nobel Prizes were awarded between 1937 and 1972, along for the work in which chromatography played a vital role. Chromatography is a physical method of separation in which the component to be separated are distributed between two phases, one of which is stationary phase while the other mobile phase which moves through it in a definite direction. The chromatographic process occur due to difference in the distribution constant of individual sample component. If we look into the definition of chromatography, so basically chromatography is a separation technique which is used to separate and identify the component of a mixture. Chromatography work by a principle by allowing the molecule present in a mixture to distribute themselves between a stationary and mobile medium. Molecules that spend most of their time in the mobile phase are carried along faster. Uh, Classification of chromatography. There are different aspects on the basis of which chromatography is classified. The first classification, which is according to the mobile phase of chromatographic technique, the first one is liquid chromatography, in which mobile phase is liquid. The second one is gas chromatography, in which mobile phase is gas. Classification on the basis of faking of the stationary phase. Uh, in this classification, the first type of chromatography is thin layer chromatography, which is also called TLC. The stationary phase is a thin layer supported on glass, plastic, or aluminum plate. Paper chromatography, in which the stationary phase is a thin film of liquid supported on inert support. The third one is column chromatography, in which stationary phase is picked in a glass column. The classification of chromatography on the basis of force of separation. So there are five types of chromatography on the basis of force of separation. The first one is adsorption chromatography, partition chromatography, ion exchange chromatography, Gill filtration chromatography, and affinity chromatography. All these types of chromatographic techniques will be discussed in detail in upcoming videos. Thank you guys for watching.